Hi, it's Alex here. Welcome to Rearing Rambles, Episode 3. I am 1 in 18. So, I bet you're wondering what that means. Let's all take a breath. Let's all relax for a sec. Breathe in and breathe out. Because we're going to talk. Today is going to be a plant episode. Because this podcast is going to be alternating between plant and animal episodes. And our last episode, episode 2, was about an animal. So now we're going to talk about plants. So, when I say I'm 1 out of 18, what I mean is I am one of the 18 people who received spores of various Lycopodiaceae plants from the American Fern Society and had successful germination. So what is the Lycopodiaceae? We're going to start there. The Lycopodiaceae is a group of fern allies. It's known as a pteridophyte. Pteridophytes are plants that are vascular, but they don't produce seeds. This includes your ferns and your fern allies. So they have a life cycle that's somewhat like that of mosses, where you have a gametophyte and sporophyte generation. Every land plant has this alternation. However, as you become more advanced and produce seed plants, the gametophyte stage is much reduced. But we're not going to worry about that today. We're going to worry about plants that produce multicellular gametophytes and that are finicky. So remember when I spoke in the pilot episode about working with ferns for educational purposes and how I was playing around with fern spores of numerous species for the purposes of finding a good species that has rapid growth and low mortality? Well, the Lycopodiaceae are a group of fern allies that are commonly known as club mosses. Sometimes they're called spike mosses. Some of them are called princess pine because they look like very miniaturized pine trees. They don't bear any close relationship to gymnosperms, but they're sometimes called princess pine. And back at the University of Rhode Island, when I was there, we noted wild populations of the Lycopodiaceae all over the place. And one of the problems was that these populations were subject to overcollection. People were collecting the rhizomes of these plants from their natural habitats and using them in wreaths and other decorations, and so it became illegal to pick them to conserve their populations. One of the interesting things about this group, as well as some other fern allies, such as Psilotum, which is the whisk fern, and we'll talk about whisk ferns in a different episode, but the spores of these plants rely on a mycorrhizal symbiont. A fungus that's naturally occurring in the soil has to actually fill that spore in order for that spore to germinate and gametophyte, and so on. Now, this presents numerous problems. It means that you can't simply take the spores and put them on any cup of soil and expect to develop them. This is the symptom that people working with orchids have had for an eternity. And so, back in the 70s, I think, it was quite a while back, a guy by the name of Dean Whittier, who unfortunately died just in 2019, began doing some experiments, along with some other people, on getting the spores of the Lycopodiaceae to germinate on artificial meat. So, tissue culture, basically. Tissue culture is the process by which you take plant material and you put it aseptically on a sterile agar that contains sugars, nutrients, and other things in order for that plant to grow. And this is a method that is used for production of virus-free plants, as well as the production of sterile plants and plants that can't normally grow outside of very tight symbiotic relationships, such as orchid seeds. And so, over the course of several decades, there were a couple of different medium types that were come up with for the production of Lycopodiaceae and some related fern allies from spores. And so, this podcast, this story, is about my adventures in going ahead and getting these spores to germinate. So we'll start with why. So, back at URI, it was my task to collect some Lycopodium and other related plants in the Lycopodiaceae to show to a class when we covered fern allies in the lab portion of the class. And I almost got killed one day because I walked through a hunting zone, firing range area looking at these things while not wearing any orange or any protection against gunshots, and people were firing their guns, and I could have very well been shot. So I don't recommend people do that sort of thing. But in any case, I had been collecting the Lycopodium, Dendrolycopodium, and other related species from these different wilderness areas in Rhode Island and Connecticut so that people could examine them. And I started thinking to myself, gee, what if URI had its own Lycopodium specimen growing out of soil so that we don't have to go out and pick pieces of these protected plants? So, I took it upon myself to obtain spores. The American Fern Society has spores of various fern ally, pteridophyte things available for members to obtain. And so I obtained four Lycopodiaceae species: Lycopodium clavatum, Dendrolycopodium dendroides, Diphasiastrum tristichium, and I believe the other one was Diphasiastrum. You know what? I'm gonna go check. I'm gonna go right now and check. I know this will be, more or less audio only, but I'm gonna quickly check what those four species were. Because I don't want to have too many interruptions to our story here. Not that. That's calanthe. That's not even a fern. Yeah, so Lycopodium clavatum. Jeez. My tissue culture box is a little disorganized. Diphasiastrum digitatum was the fourth. So it's Diphasiastrum digitatum, Diphasiastrum tristichium, Lycopodium clavatum, and Dendrolycopodium dendroides. Now, these four members of the Lycopodiaceae are not particularly unique in their requirements. They are mycorrhizal, and there was some literature out there about potential germination media for these plants. So there was an article that basically recommended that somebody use Knudsen salts with 5 grams per liter of glucose. Now, being kind of the skeptic that I am, I decided that I wanted to have more than just Knudsen medium, because Knudsen medium presents a couple of problems. One, the original formulation doesn't have any vitamins in it, or even any organic sources of nitrogen. That's a slight problem. I don't know enough about plant physiology and tissue culture to guesstimate any type of vitamins and things like that. So I decided I would get the Knudsen medium, and there are a couple of variations of Knudsen, so I bought the most unadulterated form that I could. I also obtained two other medium types, Mirashic fern medium and Parker-Thompson fern medium. And for those of you who aren't familiar with how some of this stuff works, let's just do a brief overview. Tissue culture is the process by which you take small portions of plant material and basically use a sterile medium to nourish that cell into forming more cells and eventually a new plant. There are hundreds of formulations of medium that basically provide a couple of things. They provide usually a sugar, unless you're working with something that's pretty photosynthetic on its own and doesn't need a sugar. It provides water. It provides amino acids, and it provides vitamins, trace elements, minerals, and things like that. So you're going to see common themes among different medium formulations. You're going to see nitrates. You're going to see phosphates. You're going to see potassium. You're going to see chlorides. You're going to see magnesium, manganese. You're going to see calcium in one form or another. Now, the difference is mostly what forms those chemicals are in and what their concentration is. So you can have medium that is very, very heavy in, like, I don't know, calcium nitrate, for example, where it's like every liter has like a gram of calcium nitrate. And then you can have media that doesn't use calcium nitrate, maybe it uses calcium chloride instead. And depending on the plant you're working with, one of these chemicals may be better than the other. And again, it just really all depends on what exactly you're working on. Now, so the differences between like the Parker-Thompson, the Knudsen, and the Mirashic just come down to some chemistry. And I'm not going to bore you all with like me reading the media compositions for all of these different media, just because that's not the point here. The point here is that there are multiple different formulations of tissue culture medium that one could possibly use to get the results they want with the plant tissue. And in fact, there are plenty of media that don't work for certain species while other media work just fine for those species. And so it's a big, big area, and I'm just talking about a little sliver. I'm talking about the Lycopodiaceae. And so I made the Knudsen C medium. It was just all the Knudsen salts with the glucose, the water, and the agar. And the reason I did was because that is what the literature had said. There is some scant literature out there about using Knudsen medium with glucose added to it to induce the spores of the Lycopodiaceae to germinate. Germination rates in these experiments have been very low, less than 1%. They've been very slow, taking 6 months, and there are varying reports of whether or not to pre-treat the spores by putting them on the medium and then set them in the light for a couple months before putting them in the dark. The reason they would be in the dark is because this process would happen underground, and so light would inhibit that development. But there still wasn't a lot of satisfying literature out there where somebody had worked out an exact protocol. Not to worry. I was going to do some experimentation. And, in fact, I'm still in the experimentation process. So, I made a liter of each type of medium, Knudsen C, Parker-Thompson, Mirashic, fern medium. And, all three media were the composition of the macronutrients and elements, things like that, as well as 5 grams per liter of glucose and 7 grams per liter of agar. And the medium was mixed up, autoclaved for a half hour, poured into Petri plates. Very important stuff. Then, what I did was I got these spores of these Lycopodiaceae plants that had been stored in the fridge, and the first thing I did was I put them through a vacuum pump. I put them in these glass vials with some water that had a wetting agent called tween added to it. I think I do like 6 drops of tween per liter of water, something like that. And you swirl it, you shake it, you turn on that vacuum, and after about 20 to 30 minutes, those spores sink because the water has kind of entered them, imbibed them, gotten them to sink. That's what you need first. You can't have floating spores because they're not going to be able to take up materials, and they're not going to be as easy to surface sterilize. That's important because you don't want contaminants in your cultures. So I did that, and I took my imbibed soaked spores, and I made a 0.5% sodium hypochlorite solution. The reason I say that instead of 10% bleach is because some bleach is more concentrated than 5%, so a 10% solution of some bleach might actually be higher than 0.5% sodium hypochlorite. So you'll have to adjust, like sometimes I'll have to do like a 7 or 8% bleach solution to get to the 0.5% NaOCl that I need. I soaked the spores for 15 minutes, and then I rinsed them through a sterile coffee filter with sterile water, and I dripped them onto the Petri plates of medium, sealed and labeled those Petri plates of medium, and then put them in dark boxes and kind of forgot they existed. I did this last February, so it's been about a year now. It's been roughly 51 weeks. So, what did I find? I found that the Parker-Thompson fern medium was the only medium where germination of the spores actually occurred. And I have small gametophytes of all 4 species now, with the Lycopodium and Dendrolycopodium being the quicker ones, even though they still took well over 9 months, and the 2 Diphaseastrum species being a little bit more slow. So, what we're going to do is we're going to talk now about the Parker-Thompson fern medium, some of what's already kind of known about these plants, and where to go from there. Okay. So, let's first talk about Parker-Thompson fern medium. Parker-Thompson fern medium was originally used for Ceratopterus and other ferns for use in the tissue culture of these ferns. It contains the following chemicals: ammonium molybdate, ammonium nitrate, boric acid, calcium chloride, copper sulfate pentahydrate, sodium EDTA dihydrate, ferric sulfate heptahydrate, magnesium sulfate, which is Epsom salts for people who are not aware, manganese sulfate monohydrate, potassium phosphate, and zinc sulfate heptahydrate. And this formulation was what I have found is the most effective for getting the germination and subsequent development of the spores of Lycopodiaceae. Now, this is some interesting stuff, and the reason why I'm talking about it is because there are people who are interested in propagating these plants from spores. People who want to do conservation work, people who want to do just kind of general production of these plants. And so the things that I'm curious about are, one, whether or not to try new spores on medium that has a higher concentration of glucose. Now, we're going to talk in a moment about sucrose versus glucose, but before we do, I want to mention that there hasn't really been a lot of work specifically on the Lycopodiaceae regarding sugar. The work that I'm going to be basing some of this study, future inquiry on, is actually done on Psilotum, which is whisk fern. Whisk fern is a slightly different subset of pteridiophytes that has a similar mode of growing from spore, but again, it's not exactly the same. So, in the whisk fern, what was noted is that 5 grams per liter glucose was better at in getting the spores to germinate and the gametophytes to grow quickly than 5 grams per liter of sucrose. Sucrose is table sugar. It's a more complex sugar. What was also found was that higher concentrations of sucrose led to faster growth, to the point where 40 grams per liter of sucrose, which for people who don't know anything about plant tissue culture, it's fine, you're welcome here, I love you to death. That's a very high concentration of sucrose. Most tissue culture media doesn't use more than 20 or 30 grams per liter sucrose. So we have to be open to the very distinct possibility that, perhaps that might be stressful to the plant. And indeed, with Psilotum, spore germination was slightly lower at those higher concentrations of sugar. However, gametophyte development was much more rapid. And you might be wondering, well, what's the point of getting rapid gametophyte development? A couple of things. The gametophytes are very, very vulnerable when they're really, really small. The quicker you can get them past that vulnerable point, the better off you're going to be. And also, in terms of just rapidly cycling through and getting generations in, you want as quick of a turnover as possible. And so one of the things I intend to do is use glucose at varying concentrations. Maybe I have glucose at, 10, 20, 30, and 40 grams per liter and see who develops the most rapidly on that. Because I had the 5 grams per liter just based on what was already known in the literature. So the literature out there on, the development of the spores into gametophytes of these Lycopodiaceae was done mostly for the purpose of just visualizing gametophytes and doing embryological studies. So the production aspect wasn't really paid attention to. And that's going to be an important thing because the actual things that are optimal for producing these plants in large numbers and getting higher germination rates, this and that, are not necessarily going to be found in the old literature where people just wanted to know what the gametophytes look like or how to stimulate germination in the spore. So again, the results and the intention of the study are just as important in many cases. And I'm not saying that these scientists were biased and they had a problem with these groups of plants. They just weren't as interested in, let's see how we can mass produce these, because if there's conservation reasons or people want to, apply this to horticulture and things like that, they might as well do this. That wasn't the intent. So my intent is, however, to kind of know how people can grow these things on a large scale. Because we're seeing declines in populations, we're seeing things where, these plants aren't necessarily doing as well as we'd like them to, so conservation purposes kind of demand our attention. Okay. So, I've gone over a little bit regarding the Lycopodiaceae, why the spores are special. They require in vitro tissue culture conditions to germinate and grow into your plants. Now, there are a couple of places where we still have some unknowns. For example, are there ideal media that promote the germination of the spore for the gametophyte, and then maybe a better medium for the development of the sporophyte off of the gametophytes? We don't really know. Again, this old literature is mostly concerned about visualizing the gametophytes rather than growing them on into the sporophytes. And so that presents some problems. Now, what I am planning on doing are some experiments with different glucose concentrations. Maybe I'll incorporate sucrose, but what you have to just understand is that I don't have a ton of spores right now to work with. I don't have a ton of time or space, and so I have to kind of start small. I have to, I can't set up 8 different medium types because I just don't have enough spores to go around for 8 different medium types. So what I'm probably going to start with is, a medium that has a higher concentration of glucose. See what that does. Then what I wanted to do was I wanted to get into some other chemicals to add to the medium to maybe stimulate some of that cellular division and development. So let me name a couple examples. So very classically in plant tissue culture, plant growth regulators such as auxins, cytokinins, and maybe even some others are useful in getting the proliferation of cells and development of organs and other things that you want. Because the spores start up unicellular and they're haploid, I don't know if that's the best route to take because these plant growth regulators tend to act on tissues and organs rather than single cells. With that being said, there are some compounds that we know from the orchid world that are stimulatory in the germination of seeds and development of the plants, such as peptone. I have peptone. I can play around with peptone. I can play around with calcium hydrolysate. It's an amino acid rich, it doesn't have L-glutamine, but I have supplemental L-glutamine. Amino acid rich compound that could potentially be useful for getting the spores to germinate and differentiate out. Calcium nitrate might be a good thing to play around with, and maybe even I'm forgetting what the other one was. There were several, but those are the main ones. It's calcium nitrate, peptone, calcium hydrolysate. Maybe see what having some of that in the medium will do. And that is kind of, where I'm at, where I want to, go forth and germinate these spores. And so I have a method for getting the spores to at least germinate. Once I've gotten the germinated spores and the gametophytes, then I can figure out, I have some gametophytes that look like they're getting something else on top of them, like more development. So I put those under a light to see if that would stimulate a sporophyte to appear. So I know I can do it, but the question is, how do I optimize it? Another direction I want to go in is based on a recent study done on the gametophytes of Diphasiastrum alpinum, where they actually found that it's a basidiomycete that is their fungal symbiont. And this is useful information. Now, I'm not a fungus expert, and I'm not a taxonomist in the fungi by any means, but the Basidiomycota is a large group that also includes a number of commonly cultivated, important edible mushrooms. Okay? And so what I was envisioning doing was literally growing out these mushrooms, the mycelium of these mushrooms on potato dextrose agar or light malt extract agar, and then getting the sterilized spores on a culture with these mushroom mycelia. I have 10 different Basidiomycete species right now that I'm cultivating. And whether or not it's going to work, I'm not entirely sure. But what I do know is that we can sometimes substitute types of fungi in for fungi that we want. So in the beetle and insect realm, we oftentimes make flake soil or kimchi, where we take a hardwood substrate and very rapidly break down. With flake soil, you're using yeast to ferment that wood. And with kimchi, you're using oyster mushroom to break down that wood. And these are things that these beetles wouldn't necessarily encounter in the wild, but they still do the job. And basically the role of the symbiotic fungus is to break down carbohydrates and other materials into sugars that are then fed into that spore or seed in the case of the Orchidaceae. And what we know from the Orchidaceae is that there are the fungal symbionts that you can extract from one species and actually use it to germinate some other species' seeds. And I'm not saying that it's a perfect analog. Within the Orchidaceae, you have I know that you can take Spiranthes root, macerate it, isolate the symbiotic fungus from that, and you can get seeds of some other bog orchids to germinate with it. But it doesn't mean that you're going to get, every orchid seed to germinate with that particular symbiotic fungus. And not every symbiotic fungus is going to be able to germinate multiple orchid seeds. But I figured it's still worth a shot. And the reason why is because there are some studies that suggest that when things are grown symbiotically, they're stronger and healthier, but also the fact that it might just be easier to maintain cultures of these mushrooms and put the spores in with those mushrooms. It will be a good protection against contamination, and it might just be a quicker way to actually get what you're looking for rather than using the asymbiotic medium. But I don't know. So it's definitely going to be something I look into and try. So what are some other things that may be a factor here in kind of determining how we can go about producing these fern allies? One possibility is to look into the life history of some of these plants. So if I go right now, Lycopodiaceae, let me just see. I'm trying to figure out if there's information about when these spores are released from the plant. What I'm thinking is if the spores are released in the fall, for example, a cold treatment might help. Or if the spores are released in the spring, after a certain length of time, you cold treat them. And that might keep them in sync, and that might actually stimulate some development to happen more quickly. You know, if anything, this entire process and experience has had me with more questions than answers. You know, it's not satisfying enough just to get some gametophytes to appear. It's going to be satisfying when I have a more optimal way of getting these spores to germinate, produce the gametophytes, produce the sporophytes. Because we've just scratched the surface. We've just figured out how to get these gametophytes to appear from the spores, how to germinate the spores. That's not the end. That's only half the battle. The other half is going to be really figuring out how we're going to get the sporophytes to appear, acclimate them to ex vitro conditions, grow them on, and just keep them going. And so I encourage people to not stop trying. Always ask questions. Keep going. We are going to figure this out. This is going to be something that, 10 years from now isn't going to be a problem. We're going to know how to germinate these things from spores. We're going to know enough about their biology. And it just starts with, a little bit of success, a little bit of encouragement, and then people asking the right questions and running the right experiments. And so, for those of you interested in growing Lycopodiaceae from spores, my suggestion is to start with Parker-Thompson fern medium. Probably want to have 10 to 15 grams per liter of glucose added to that. You're going to want to have those spores imbibe. You're going to want to bleach those spores for, 15 minutes. You're going to want to sow them. You're going to want to keep them in the dark, room temperature. And, maybe from there we do experiments on other things to add to the medium. Maybe from there we do experiments on, like, thermal timing or, light shock treatments, where we keep them under a light for a couple of weeks first and see where we can get as far as this goes. So this is a short one, not my normal hour-long podcast, but I want to thank everybody for tuning in and listening, and I hope that everybody, stays curious about the natural world and, wants to know more and wants to grow things and have fun. This has been I Am One of 18. Thank you for joining me.